Hi, class. In this recording, we're going to talk about the blood-brain barrier. So, as we're looking at our blood-brain barrier, this is a pretty big issue in our brain. The blood-brain barrier is going to help to control what gets into and gets out of the brain. It helps to regulate what makes it into, excuse me, the cerebral spinal fluid as well. As we're looking at our brain, it's about 2% of our adult mass of our body. It receives about 15% of the overall blood supply of our bodies, or about 3/4 of a liter per minute. So our brain is disproportionately important in our body. It's kind of like the United States of our body, and then it uses up many more resources relative to other parts of its body, our body, relative to the available resources. And as we look at our brain, the neurons in particular of the brain have a very high demand for ATP. Therefore, they have a lot of demand for oxygen and glucose. Even though the ependymal cells, in the last recording we made, I had mentioned how ependymal cells down-regulate the amount of glucose that's present in the cerebral spinal fluid, we still do need to have a good supply of glucose in the cerebral spinal fluid. So as we look at the brain, if we have 10 seconds of blood flow being denied to the brain, that can cause loss of consciousness. If you've ever been put in a sleeper hold, you know exactly what I'm talking about, where somebody can just squeeze your left, right carotid arteries and knock you out in a very short period of time. If that pressure is applied for 1 to 2 minutes, there can be significant impairment, aka there can be a loss of function or reduction of function in the brain. And if they go 4 minutes without blood supply, there can be irreversible brain damage which occurs. Not cool at all. So as we look at our brain is going to have the blood-brain barrier system that regulates and tightly controls what goes from the blood into the brain. And one of the reasons we need to tightly control this is because the blood can oftentimes be a vehicle, a method to take a pathogen from the surface of our body and transmit all throughout the rest of our body. It's kind of the interstate system or the 8-lane superhighway of our body. If we get a cut or a scratch, a pathogen can enter the blood, and we don't want that pathogen getting into the brain. So as we look at the brain, we need to tightly guard access to the brain. The blood capillaries throughout the brain tissue are going to be a point of access. And then we also are going to have the blood capillaries of the choroid plexus as another point of access to get into the brain. These are all going to be guarded by that blood-brain barrier system. So as we're looking at the blood-brain barrier system, it's going to consist of tight junctions in between the cells of the capillaries. So in a regular capillary, the blood plasma can ooze right in between those cells that line the capillary, those endothelial cells. Within the blood-brain barrier system, instead of having those gaps, we are instead going to have tight junctions in between the blood capillaries. And those tight junctions are going to be stimulated to develop by the glial cells of the brain, in particular the astrocytes of the brain, reaching out their pseudopods and wrapping them around the blood-brain capillaries, stimulating the development of the tight junctions. And then those tight junctions mean that anything that leaves the blood supply has to pass through cells. Those substances need to pass through the blood capillary cells, those endothelial cells directly. And that helps to downregulate the amount of pathogens and harmful substances which will access the brain. Now, as we're looking at the blood-brain cerebral spinal fluid barrier, it's going to have a lot of junctions present between the ependymal cells of the blood-brain barrier. Then these tight junctions are going to be absent from ependymal cells, which are elsewhere in the brain. And this allows for there to be an exchange between the brain tissue, fluid exchange between brain tissue and the cerebral spinal fluid. As we look at the blood-brain barrier system, it's going to be highly permeable to a few select things. So let's go all the way back to chapter 2. In chapter 2, we talked about chemistry and how like dissolves like. And if we look at the cell membrane, it's made of phospholipids, a phospholipid bilayer, and those fatty acid tails are very hydrophobic, or they are neutral or non-polar. So if we're looking at small substances that are neutral, like oxygen or carbon dioxide, because carbon dioxide has equal electron distribution across its surface. These substances can go straight through the blood-brain barrier. Other substances that have nonpolar regions, like alcohol, caffeine, and nicotine, can go right through the blood-brain barrier. Water is small enough, even though it's highly polar, it can go through the blood-brain barrier. Glucose is going to have a lot of transporters that allow it to get through the blood-brain barrier. Anesthetics, when designing an anesthetic or analgesic medication, this anesthetic needs to be designed in such a way that it's a small molecule with a nonpolar region. Typically, it's going to be a Zwitter ion or amphipathic, so it can have a polar and nonpolar region, and that will allow it to go through the blood-brain barrier. Our blood-brain barrier is slightly soluble to sodium, potassium, chloride, so these highly charged ionic compounds or ions can slightly make it through the blood-brain barrier just because they are unbelievably small molecules. Generally speaking, to really paraphrase, something can get to the blood-brain barrier if it's really, really tiny or if it has a non-polar region on its molecular structure. Now, as we're looking at the blood-brain barrier system, it's a big problem if we want to get a medication into the central nervous system. The blood-brain barrier was a topic of research for my wife, and she spent a lot of time working in the lab trying to synthesize and deduce the structure of the protein within some of the proteins within the blood-brain barrier. This is one of the reasons why when we deliver medications to the central nervous system, a lot of times those medications need to be delivered via spinal tap. So we can bypass the meninges and get that medication directly into the cerebral spinal fluid to help upregulate its absorption into the brain tissue. If we have trauma or inflammation into the brain, that can damage the blood-brain barrier system and allow pathogens to enter the brain. We also have some circumventricular organs. There are some places within the third and fourth ventricle where there's no blood-brain barrier at all. Our brain, the neurons of our brain, do need to access and monitor blood chemistry and blood quality directly to help regulate our body's ability to have homeostasis. So we need our blood to have direct access to the brain for a few specific reasons. We need to respond to fluctuations in blood glucose, pH, osmolarity, blood oxygen, blood carbon dioxide, et cetera, et cetera. So in these situations where we have a dendrite of a neuron being exposed directly to the bloodstream to monitor blood chemistry, these circumventricular organs are going to be a route for pathogens to get into the brain directly because they can infect the neurons of the central nervous system because the neuron has so to speak, 1 foot outside of the central nervous system, or a dendrite or two outside of the central nervous system that's open to attack. That's all we have for this recording on the blood-brain barrier system. If you have any questions, please feel free to post them in the class discussion board or shoot me an email. And as always, happy studies.